Yeah, my name is Thomas Anderson. I guess my talk, I'm going to do some background. Who am I? What is microdosing? Talk about methods and talk about uh, the research that we've done. I'll talk about the code book that we made. My understanding is that you guys have had some articles sent your way, and then I'll talk about open science as well. And I'm going to try and mostly breeze through relatively quickly because my goal here is to have more time for questions, and I want to do that. So who am I? My name is Thomas Anderson. I'm a PhD candidate in Norman Farb's lab at the University of Toronto. I am a cognitive neuroscientist by trade. That's my focus of my PhD, is actually on meta-awareness, which is sort of a facet of attention, sort of how you notice that your own mind is wandering. I'm also, but why am I talking with you? So I've also published one of the first studies on microdosing psychedelics. We published, one of the first in psychopharmacology based on a survey that we did on Reddit. And then we published another one. This is the Psychedelic Microdosing Benefits and Challenges Codebook that we'll be talking about today in more depth. We also published a third based on that same sample about psychiatric comorbidities and demographics of people who microdose. We've also submitted 3 other papers on, one of them is the Global Drug Survey Microdosing Questions, which has the largest microdosing sample to date. It's almost 8,000 microdosers. Another one is talking about microdosing and the meaning crisis, which is sort of a more qualitative look at why people are microdosing, what they get out of it, or what they claim to get out of it. And another paper that was the other one that was sent out, I believe, that talks about the need for transparency in research and the importance of open science. I have also co-founded the Psychedelic Studies Research Program at U of T. I'm the research director there. And with my partner, Rotem Petranker, we started a psychedelic consulting company because people wanted to give us money, and U of T was a bit of a hassle to work with. So we wanted to find a different way to be able to bring money in to U of T to do this research without having to always talk with their people first. People were approaching us, and they wanted to talk to us. They didn't want to be handed off to someone else. So that's sort of my background. What is this thing we call microdosing? So I'm going to assume some familiarity with psychedelics, but microdosing is basically the practice of ingesting sub-hallucinogenic doses of a psychedelic, and that's usually about 10% of a higher dose of a higher, like, recreational level dose or clinical dose. So for LSD, that might be 10% of a tab instead of a full tab, maybe 10 to 20 micrograms. And for psilocybin, usually consumed in the form of dried mushrooms, that would be 0.2 to 0.3 grams of the dried mushrooms themselves. We've got a lot of anecdotal evidence that people are interested in microdosing, but we wanted to bring a more rigorous scientific approach to the study of microdosing, and there wasn't any research at all in the field, so we decided to do some. We started it with a survey, mostly on Reddit, on a variety of subreddits, but also shared through Facebook and Twitter and whatever other social medias. And what I'll be talking about today, or what I'll be focusing on today, is the codebook part. So to do this codebook, we use what is called the grounded theory method. So we find this great population of people. So we have, the r/microdosing subreddit is now at over 100,000 subscribers. So people are talking about this, and there's very little research. So we had this great sample of people who have microdosed, and we wanted to get information from them. So we solicited reports from them in our survey. I think for this particular sample, we had 278 responses. In our broader survey, we had 909 respondents. And I can talk about some of those results if people have questions. But for this stuff, we actually had 278 microdosers. We get their reports, then we analyze them. We look at each report that a person gives, and then we abstract those to different levels of abstraction so that we can then create sort of a summary list of categories of benefits and challenges that people who have microdosed report having or report being associated with microdosing. So to get really specific, this is what our survey looked like. We asked people about the benefits and challenges or drawbacks and the pros and cons of microdosing. People could then fill in they would fill in their first benefit, second benefit, third benefit, and then they would rate the importance of the benefits on whatever on their subjective scale here. Then people would also do the same thing for drawbacks. Then we get back those raw answers. So whenever people fill in that first benefit, second benefit, we get those back as these raw answers. And so you see focus, antidepressant effects, antidepressant mood lift, awareness, happiness, focus, and so on. But what we then do to analyze that is you maybe already see a pattern here that these seem to fit together as something that are describing the same thing. So when people are describing the same thing, we then note that down, we code it in the particular way. So you see when people say focus, we write improved focus, and then those become a collection of codes, individual codes, and we have a bunch of those, and then we group the codes together into subcategories, and then we group those categories into the broader category. And then we're able to talk about categories of benefit and categories of challenge associated with microdosing. We're able to sum them up under the categories and fit them all together and talk about potential research avenues. Because of course, this is all people reporting on their subjective experience of microdosing. These are not, proven effects that we've seen in randomized placebo-controlled trials. None of that is the case. This is all just people reporting, confounded entirely with placebo effect and all of the other possible confounds. But this is what we get. So we get these challenges, the benefits and challenges of microdosing. And so on here, you'll see some of the most prevalent ones. On the benefit side, we see improved mood is a big one. We see improved focus, and we see creativity and self-efficacy. And then on the challenges side, we see illegality is the main big one. And then we also see physiological discomfort. So in this case, illegality has a lot to do with, microdosing is illegal. It's not really an outcome of microdosing. It's more of the cultural context in which microdosing takes place creates challenges. And then there's also the stigma associated with microdosing and problems that come with trying to dose properly because these are unregulated substances. You can imagine if you wanted to take a Tylenol, but every time you went to the pharmacy to buy Tylenol, the dose was always different, then you might have some trouble dosing. And in Tylenol's case, you might damage your liver if you took too much. But in the psychedelic case, you might accidentally take too much of a microdose, and then it's no longer a microdose. You're having sort of a small trip. And then we also saw physiological discomfort. That tends to be temperature dysregulation problems. So things just like feeling a little too hot, feeling too cold, clammy, gastrointestinal stuff, mushrooms taste bad. Some people have sort of a nauseous response to that sort of thing. So one of the other things we can see on here is that there are a lot of parallels between the benefits and the challenges. So that was one of the things we figured out going through these, that they're all paralleled except for creativity and illegality. So no one was saying, man, one of the really tough things about microdosing is that it makes me less creative. No one is saying that. On the other side, no one is saying one of the great things about microdosing is how illegal it is. No one is saying that either. So we see that these are sort of special cases, but for the most part, we see potential benefits and we also see potential downfalls. So one thing we wanted to do with this study is think about where we could take this in the future. And so we created this list by taking the difference between the benefits and the challenges. And we see that we have sort of isolated mood, self-focus, or mood, self-efficacy, and focus as being highest potential research avenues for microdosing, in our opinion. And then other physiological symptoms, those kinds of things, as potential drawbacks to microdosing that we want to keep a watch out for. When we recently worked with the Global Drug Survey, we used our benefits and challenges model, and we tested in this huge sample of almost 8,000 microdosers, again, what the most prevalent, whether people actually experience these different categories. And we did see that mood was, again, the most common benefit that people reported from microdosing. And also creativity was way up there. And we did also see actually that none was the most common drawback or challenge associated with microdosing, which was really interesting to see that people were saying that there really wasn't anything, but a fair amount of people also said physiological concerns and this impairment of focus. So I think I'm running quickly enough. So then the next thing I would want to talk about is open science. So that is sort of the first paper covered, the code book, and then we'll have absolutely lots of time for questions. And then this open science bit. So I'll kind of run into this quickly. One of the things that our group is doing differently that we find really important is open science. So why would we find open science to be that important? What's the problem that we're trying to address here? Basically, there's this thing called the replicability or reproducibility crisis in science. Those are two very closely related things, where in science, really the main thing that you're supposed to be able to do is repeat an experiment and get the same results. So if you, if an apple falls on Newton's head, then an apple should fall on someone else's head in the same way and shouldn't like float away and be something different. The problem is in fields like psychology, especially, and especially social psychology, but also medicine and biology, this isn't happening. This isn't happening properly. And about, depending on the particular metric you use, maybe half of all of psychology or more doesn't replicate. So this is a case where there are lots of problems. There's lots of factors that contribute to why this is a problem: selective reporting, presenting your data in this way that is called harking, which is where you collect your data, you run your analysis, and then you write your hypotheses after you already know the answer. That's not how science is actually supposed to be done. That is completely broken, but people have been doing that, and it's caused a whole bunch of stuff to not work properly because they don't understand statistics or how statistics works. So we get a lot of garbage science that doesn't replicate. That's a problem. It's a waste of everyone's time. It's a waste of money, a waste of public funding, and it also hurts trust in science whenever science doesn't actually work. So the solution to this, we believe, is open science, which is pre-registering your plan, sharing your data, and sharing your materials. So when we're talking about pre-registration, this is where you design your study, then you pre-register, which is basically just saying you report in a place that is time-stamped and will become public what you are planning to do, and that's it. You just write down what you're planning to do, and then you register that. Then you do what you plan to do, and you can run exploratory analysis. That's totally cool. But then once you're done, you publish. And then everyone can see what you plan to do versus what you actually did. And that's the whole goal of pre-registering, is that it holds you accountable to your previous self of what you were planning to do. There are a variety of ways of pre-registering that I won't really go into right now, but there are different levels of structure, and generally our group prefers the most structured method, which requires you to report the most detail about what you're going to do, including statistical analyses, sample size rules, stopping rules, how you're going to exclude data. There are so many researcher degrees of freedom, which is basically little decisions that researchers can make that can end up fudging how the results turn out. And we want to lock those down, and we don't want to fudge anything. We want everything to be as rigorous and transparent as possible. So we prefer the most structured method. There's also sharing data and materials. So it is actually super easy these days to share your data and share your materials. Like I shared these slides before I began on ResearchGate. It's that easy. We live in a world of technology where it's super easy to share everything. And open data and open materials is whenever you take all the data you collect and then you put it somewhere where everyone else can look at it, everyone else can make sure that it's good, they can check the quality, they can explore it on their own, they can check your own statistics and that sort of thing. So the research that I was talking about before, from the codebook paper, all of that is available. Anyone, scientist, lay scientist, lay person with no background in anything, everyone can go and look it up and find the data and do whatever they want with it, because that's open data. And then open materials is sharing the way that you got your data. So questionnaires that you asked, any kind of transformations that you do, any tasks that you do, code that you use, that sort of thing. So that's sort of where I'm coming from. And of course, I want to thank Rotem Petranker, who is my colleague and business partner in this, Norman Farb, my supervisor, and the Nikian Foundation. So they have actually provided us with $350,000 to run the first clinical trial, phase 2 clinical trial on microdosing psilocybin, which is what we're planning to do. Our application is pretty much ready, and then we were kind of planning to get going this summer, but as you can imagine with the whole COVID thing, we're not really ready to do that anymore, but we're sort of ready to hit the ground running and send our stuff to Health Canada whenever we get the chance. In terms of connecting, there are some websites there you can find me at. By all means, feel free to email me, but I do have a request. Try not to email me if you want to participate in our studies. We're not running any studies right now. I have nothing to do with your email. If you have health issues and you want psychedelics, I can't give you psychedelics. Or if you want psychedelic psychotherapy, I also can't do that for you. I'm a cognitive neuroscientist. I can't actually help you with that. And there's no legal way for me to help you with that anyway. I would suggest getting in touch with your local psychedelic society or psychedelic community and talking to people who are in that area and are interested in the same things that you're interested in. But I personally cannot help you. But feel free to email me, especially if you're an academic and you want to collaborate, or if you're a philanthropist and you want to give us money. That would be, we love money. You know, everyone loves money. Everything needs money to work properly. So that's me. Leo, if you have, I hope that was fast enough and kind of sped through it, but I hope that there are some questions that we can take.